Hello students, I'm Madam Joyce from Educare TZ. I'm teaching biology and today I'll be with you to discuss the topic that is the cytology, whereby it is found in Form 5. Here we are going to focus dealing with our subtopic, which is the biochemistry. So in the biochemistry we tend to discuss the organic constituents which are found within the cell and its function and how are they formed. So in the previous session we terminate by looking on the organic constituent in terms of the function, that is the lipid. So in this session we are going to see another organic constituent which is found within the cell, and this is the proteins. So now simply we can say that the proteins are large molecules or macromolecules consisting of one or more long chain of amino acid. So his protein tunasema kwamba they are large biomolecules. So we can say biomolecule, or it is a macromolecule. Macromolecule. They are essential molecules which are found within the body of living organism. But these protein, they consist one or more long chain of amino acid. So simply we can say that proteins, they are made with various or variety of amino acid and thus tend to form a chain. And now we can simply also say that a molecule contains carbon, hydrogen, oxygen, nitrogen, sulfur and some phosphorus. So ile molecule ya protein mpaka inaenda kutengeneza chain, tunasema kwamba imetengenezwa na about 1 2 3 4 5 6 molecule or 6 atoms or 6 elements. And the first element here, we have carbon, we have hydrogen, we have oxygen, the nitrogen, sulfur and some phosphorus. So now we can say that the amino group provide protein's basic structure, and the carbonyl group give the acidic nature of the amino acid. So tunaweza tukasema kwamba hii protein inakuwa kwenye structure yake imebeba base structure pamoja na protein, the acid structure. So the base structure, it comes from the amino group, that is NH2. That is the base structure of a protein. And the carbonyl group or carboxyl group, that is a COOH, which is an acid. So kwenye structure ya hile ya protein kuna basic structure na kuna acidic structure. You have to know all of them. I have said that the acidic structure, it comes from the amino group, and the acidic structure comes from the carbonyl group, and the basic structure comes from the amino group. So the other group is known as the side chain, which represent the hydrogen atom or any other group such as the alkyl group. We're going to see that in the figure 1.31. Kwa maana kwamba kwenye hile structure ya protein kuna ya structure ambayo tumeona ni amino group, kuna structure ya carboxyl group, na pia kuna structure ambayo imeandikwa R group. Sasa hii hapa R ina stand for hydrogen atom, au inaweza ikawa any other alkyl group, that is inaweza ikawa methyl, inaweza ikawa ethyl, inaweza ikawa pentyl, inaweza ikawa anything that is alkyl, alkane, inaweza ikawa anything, methane, that is alkyl. So when you have been asked to explain the structure, you have to say that the structure of the protein, it has been made with 3 parts, or it has been divided into 3 parts. In the first part, we have already seen it is the amino group. The second part, it is the carboxyl group. And the last part, it is ARA group. So the proteins differ from one another primarily in their sequence of amino acid, which is detected by the nucleotide sequence of their genes, and which usually result in the protein folding and specific three-dimensional structure that determine its activity. So ile structure ya proteins tumesema kwamba imetengenezwa na amino acid. And imetengenezwa na amino acid. And this amino acid, they tend to be detected by the nucleotide sequence. This is how is it formed. It is formed there in the DNA and so on. So now in the nucleotide sequence of their genes, and which usually result in the protein folding into specific three-dimensional structure that determine its activity. And this one is the figure 1.31, which shows the structure of the amino acid. So now you can see within the structure of amino acid zikicombine, yani zikiwa nyingi, ndipo ambapo inatengeneza chain of a protein. So hii structure ya amino acid imetengenezwa na nini? Structure ya amino acid imetengenezwa na amino group ambayo ndio hii hapa NH2. That is hydrogen ya kwanza hiyo na hydrogen ya pili ndio ambayo ina bring about that NH2. We have also the another part which is it is found at the middle, that is the side chain, which is ARA group. Tulisema kwamba hii ARA group inaweza ikawa ni hydrogen atom au inaweza ikawa any alkyl group. So tukiongelea alkyl group hapa tunaweza tukasema kwamba inaweza ikawa pentane, inaweza ikawa alkene, inaweza ikawa alkyne and so on. Those one, they are alkyl group. But also we have the last part, which is the carboxylic acid. Which is the carboxylic acid. So now you can see COOH. This one is oxygen atom and this one is oxygen atom and one hydrogen atom that bring about carboxylic acid in nature. Remember, while we are discussing on the structure of protein, we have seen also that the protein, it has basic structure and acidic structure. This part contributed to basic structure, and this part it contributed to the acidic structure. So you're going to see later more on the structure which consists of acid and basic structure. Why is it called? Oh, it is known as what? We're going to see later on. So now first let's see the properties of the proteins. And the first property of protein, they are colorless. The proteins, they are colorless. So the crystalline solid, which are soluble in water but insoluble in organic solvents such as the ether, chloroforms, and the acetones. So these molecule of proteins which have been made with a long chain of amino acid, we have seen that they are colorless compared to the rest of the organic molecule, that is carbohydrate and lipid. The next property of a lipid, we can say that they are amphoteric compounds, as they have both acidic and basic property. This is what I've told you we're going to learn in this in this slide. That is amphoteric compound. The proteins, they are amphoteric in nature. Why are they called amphoteric in nature? The protein, they are amphoteric in nature since they have both property of acidic and basic property. The acidic property, it comes from carboxylic acid group, but the basic property of a protein, they come from the amino acid, and thus bring about the amphoteric nature or the amphoteric compound. These are the proteins. We have another property of protein, that is protein in neutral aqueous solution. They exist as a dipolar or zwitter ions. On one side, the acidic carboxylic group has a tendency of donating hydrogen ion atom. So the proteins in aqueous media, they can exist as a dipolar or they can exist as a zwitter ions, meaning that they tend to have ions which is hanging on their structure. That ions, it has a tendency to donate hydrogen ion or protons. You're going to discuss in detail about this zwitter ion or zwitter nature or zwitter ion nature, which is found in protein. So here you have only to know that they are protein in aqueous media or in aqueous solution. They tend to have, or they exist as a dipolar or zwitter ions. Why? Because there is a presence of hydrogen donating atom or donating hydrogen. So they tend to donate a pair of hydrogen atom. We have another property of lipid. So let's explain about this zwitter nature of protein. So now you can see, therefore, it dissociates to release hydrogen ion and become negatively charged. So on the other side, the basic amino group has high affinity to hydrogen, therefore it accepts hydrogen ion and become positively charged. So the zwitter ions, which are found in a proteins, that is, it tend to have one loosely hydrogen atom. So hapa tunasema kwamba ile hydrogen atom tend to be released, itatolewa kwenye ile structure ya proteins. Ikishatolewa, ile compound nzima itakuwa negatively charged. Thereby on the other side, which is in the basic solution or in the basic compound, now you are going to see it has high affinity to hydrogen. Therefore, it tends to accept the hydrogen ion and become positively charged kwenye ile structure. Kwa ina maana ile loosely hydrogen inatoka kwenye acid inaenda kwenye ile basic structure of the basic structure of that lipid. So now you can see each amino acid has its own pH value at which it exists in its neutral zwitter ion form. So this pH at which the amino acid are electrically neutral is termed as isoelectric point. My student, for more clarification on the zwitter ions which are found within the protein, I would like to invite you into the next part of this session, whereby I will going to clarify more about this zwitter or dipolar nature of a protein. So thank you for your time and for listening.